Okay, good morning. Thank you all for being here this morning. We're going to get started right now. The first thing I'm going to say is if you have not already done so, please either scan the QR code or go to menti.com and enter the code. We have some questions we're going to be asking you throughout the day, and that'll let you access the answers. So welcome. My name is Monica Wirt-Parkinson. I'm the Director of Community Engagement, and we're so happy to have you here. We have not had students back to campus like this since 2020. So welcome back. We're so happy to have you. Okay. Before we get started today, I first wanted to thank our sponsors. First is the March of Dimes. Has anyone in here heard of the March of Dimes before? Okay, great. I'm so glad to hear that. They are one of the founding donors of the Salk Institute and still continue to make programs like this possible. So a huge thank you to the March of Dimes. And also a huge thank you to the Blue family. It's thanks to their continued support that we've been able to offer this particular program, and they're actually here with us today. So we wanted to say hi and welcome, and if you can give them a great round of applause. I also want to say thank you to all of our scientists and employees and volunteers that are here today. They're going to be leading you through the labs and leading you through workshops, and we're so grateful that they gave up their Saturday morning to be here with us to lead you through this great experience. Okay. So, as you may have figured out by now, Salk is not an acronym. It actually stands for Dr. Jonas Salk. Has anyone here heard of Jonas Salk before? Okay. Have you heard of him recently because of all the news and the talk about the pandemic and COVID-19 in the news? Is that how you've heard of him, or did you hear of him before that? Before? Oh, great. Okay, good. So Dr. Jonas Salk, and this is him right here, he's sitting in the courtyard, which is normally very beautiful and not full of rain, and he is watching the sun setting twice a year. On the solstice, the sun sets exactly dead center down the middle of the courtyard. And Jonas Salk was really important in the 40s and the 50s because during the polio epidemic, it was not a pandemic, it was an epidemic, he actually developed the world's first safe and effective polio vaccine. And then when he developed the vaccine, what he did was something pretty novel, is he decided not to patent it. He did not make any money off the polio vaccine. So what he wanted to do instead was develop an institution where scientists had complete freedom to understand the mechanisms of the body and to make the world a better place. And so you're actually standing in his dream right now. This is the Salk Institute. And as I mentioned before, it was founded with seed money from the March of Dimes, and the citizens of San Diego actually voted to give Jonas Salk this land to build his institute on. So these two great things came together at the same time, and we were able to build the Salk Institute. One of the things that Jonas Salk was really, really passionate about was training the next generation of scientists. And one of his most famous quotes is, "Our greatest responsibility is to be good ancestors." And it was through his vision and his passion that we were able to found these education outreach programs today. He actually was one of the founding members and supportive members of this program, and he really believed it was important to inspire students like you to consider careers in science. So now, 60 years later, here we are. We're here at the Salk Institute. And what we are is a basic biological research facility. That means we're not a hospital, we're not a degree-granting institution. What we do is we really work to understand the fundamental mechanisms of the body and human health. Our scientists, we've had over 6 Nobel laureates here, we've had 39 biotech companies that have spun off, we have 54 countries represented by our scientific staff, and we hold over 580 US and foreign patents. So for a really small organization, we've been pretty mighty in how we've been able to impact science. So what do scientists at a basic biological research institute study? So we work to understand the basic mechanisms of the body that can give rise to disease. And one of those diseases that we study a lot is cancer. Even in recent decades, cancer has been something that we don't really understand how it works. So our scientists are working to understand basic biological research that gives rise to the mechanisms of cancer. And in fact, since the very beginning of the Salk Institute, we've been one of the powerhouses of cancer research. We were one of the first basic research institutes designated as an NCI, a National Cancer Institute. And that's something we've held for over 60 years. So that's something we're really proud of, that we're one of the powerhouses of understanding basic fundamental mechanisms of cancer that will then give rise to therapeutics. We also work to understand aging. There's a lot of molecular and cellular causes of aging, and we're working for ways to stave off diseases like Alzheimer's, diabetes, cancer, cardiovascular disease, and other age-related ailments. So we're not really trying to make people live longer. We're just trying to make sure that as they're alive, they have a healthier lifespan. So you stay healthier longer. That's one of our goals. We also study plants. And in fact, we're one of the top plant biology research institutes in the world. One of our PIs, Dr. Joanne Corey, is pretty much the most amazing plant researcher that you're going to find in the world. And it was through her efforts and the efforts of our other plant scientists that we now have an initiative called Harnessing Plants. And what we're trying to do is combat climate change. And we're trying to do that by using plants, which already naturally sequester carbon in their roots. We're trying to teach them to do that a little bit better. The longer and deeper you can grow a root, the more carbon that you can store and pull out of the atmosphere. So we're trying to use a natural cycle of the earth to combat climate change. And we're using that through a couple of different initiatives. So all of this amazing research on campus is supported through something we call core facilities. And you're very lucky because you're going to go through a couple of cores today, and you're actually going to hear next from one of our core directors. So science can be very expensive. You need a lot of cutting-edge technology. And one of the really cool things that the Salk Institute does is we create cores that are centralized areas of really high technological advances, and they're run by experts in their field. So all of our researchers across campus are able to access these cores and use the technology that's in them. So we're all here today because Jonah Salk had a vision of education outreach. And today that's turned into our community engagement department. And we have a mission, which is teaching, inspiring, and promoting. So all of these programs, like the one you're at today, are seen through this lens of teaching, inspiring, and promoting. And that's something that our team, which I'll bring up right now, all works on. So we're a really small but mighty group of professionals that work together to ensure that Jonah Salk's vision for the future is staying true. And I would like to point out Shoshana. She's our fabulous team member who you probably all saw coming in. It's thanks to her that this day is happening. So let's give Shoshana right here in the front a round of applause. We really appreciate everything she does. And she's supported by the rest of our fabulous team members who you probably have all seen here today. So not only do we organize this fantastic high school science day, we have a couple of other programs that we also put on. Two of the ones that are probably most interesting to you are our side chats at Salk. We can Zoom a scientist directly into your classroom to talk about their research and answer questions. We also have a high school internship program. How many of you have heard of our Toff Brody High School Summer Scholars program? Has anyone here heard of it? Okay, good. Fantastic. So the applications are open right now. That's why I'm telling you about it. They're open through April 7th. You have a chance to work for 8 weeks in a lab working on a direct research project. It is paid. You receive a 1-week boot camp training, and you learn how to put together a scientific presentation at the end of those 8 weeks. It is an amazing program. So if you're at all interested in seeing what it's like to work in a lab, I definitely encourage you to apply. Applications are open now through April 7th. We will be offering a couple of different application workshops, so you can check back on our website for details of when you can sign up for one of those, but definitely check that program out. A couple of other programs that we offer, and this may be more interesting to your teachers in the group, is we have a symposium for teachers. What we do is we bring them in and pair them directly with our researchers so they can learn about real-life research happening in the lab, and then we help them develop lesson plans so they can take that back to the classroom and teach you about some of the research at Salk. Our side chats at Salk, I mentioned earlier, that is our virtual program. We Zoom a scientist into the classroom, and they tell you a little bit about their research, how they became a scientist, and we have topics that we can offer on everything from plant biology to cancer research to neuroscience. So if there's something of interest to you, we can definitely Zoom a scientist into your classroom. And finally, Explore Salk. It's a free community open house that we offer. We have not been able to offer it since 2019, so we're really excited. Save the date for October 28th. That is Jonah Salk's birthday, and we will be opening the campus and inviting anyone who wants to come on to hear from our researchers, do hands-on research, and attend science talks. Okay, thank you for listening through my introduction. Now we get to the really exciting part of the program, which is hearing directly from our guest scientist speaker this morning. We are going to be joined by Dr. Elsa Molina. As I mentioned, she is the director of the Next Generation Sequencing Corps, and she's going to join us right now and tell us a little bit more about her journey to become a scientist and the research that she's working on right now. So please join me in welcoming Dr. Molina to the podium.
Students, if some of you have not scanned the QR code for Mentimeter or gone to our website yet, please put that in right now. We have a question live at the moment. So go to menti.com and put in 74715626 or scan the QR code in the corner. We're going to be asking questions to you throughout Elsa's talk, and you also are able to submit questions anonymously through that app. So please scan the QR code. I'm going to leave it up for just a couple more seconds, and then we'll let Elsa take it away. Okay, does anyone still need it?
I would like to thank you very much, Monica, for the presentation, and I would like also to thank very much the Education Outreach Committee who invited me today here. So welcome everyone, welcome at the Salk Institute. My name is Dr. Elsa Molina. I'm the director of the Next Generation Sequencing Corps here, and today it's a pleasure to tell you about genomics and how I fell in love with it. So first, right now, we are at the Salk Institute, and I am at the Salk Institute. And if you would have told me at your age that I will be here at the Salk, you know, many years later, I would have told you, no, it's impossible, I don't trust you. And here I am, you know. So I will tell you how during my background, I had several opportunities that ended up me here, actually. So this slide is to tell you, like, trust in yourself, and please take all the opportunities that you have in your life, because you might ended up in a place that you really like, and the most important things I think in your life is like to like what you are doing every day, and you like what you want to be, you know. So at your age, I was asked, you know, by the teachers, by your parents, or what you want to be later in your life. And this question might be very stressful. So at your age, I was quite stressed by this question, like, oh my God, what I want to do, which, because you are at school, so you like some subjects, but sometimes you like the class, but you don't get a good mark. So all these questions that you have in your head and all the questions that your teacher asks you, it can be very stressful. So some people, like today, apparently, according to the question that you just answered, some people know already what they would like to be in the future. So that's great. But some people are still struggling. So just don't be too stressed. It happened to a lot of people. At your age, I didn't know that I would be like a scientist today, and I would be very happy to be a scientist. So I will tell you a little bit of my background. So as you can hear, I'm French, so I was born and raised in France, in a city called Bourges, that is 2 hours in the south of Paris. So it's a kind of a countryside, and just after high school, I had to decide where to go to college. And it was very stressful for me because I didn't know if I wanted to go to a college and take like a literature class or mathematics class or biology. I didn't really know, because during all my scholar courses, I was sometimes like very good in math, sometimes not. I was very good in biology, sometimes not. So it was kind of like stressful for me. And so I've decided, okay, what I like to study, what I like to listen to, when the teachers is doing the class, what I like to listen to, what I like to in my life. So I've decided that, okay, I think I really like biology. And then I had to decide where to go, because in France there are several universities where you can study biology. And what I know is I didn't want to study biology in my hometown. For some reason, I really like traveling, as you are going to see, and I decided to go in a city where I didn't have friends, I didn't have family, it was far away from my home, that this is perfect. So I ended up at the University of Limoges. That is 2 hours and a half of driving from my hometown, without traffic, because in France there is not much traffic, it's not like here. And it was like fantastic. I really had great time doing my bachelor there. And in France, when you start your bachelor in biology, it's quite general. So you have geology, optics, physics, chemistry, biochemistry, physiology. You have all these things the first year. So that can be challenging, because for some people that do not like geology, they have to pass to go to the second year. So it was quite challenging for me, because I figured out that I didn't like at all physics, optics, fluids, and all that stuff. But I had to pass to go to the second year. And then I had my bachelor degree in cell biology, physiology, and immunology. And during my master, so in France, the master is 2 years. So you have 1 year master, second year master. During my first year of my master at the University of Limoges, we had this opportunity to do an internship, a 2-month internship, wherever we wanted. And I was like, oh, I really want to see the world and travel. So I basically went on the web page where we had all the the partnerships universities in the world. And I was like, okay, where I want to go for this 2-month internship, because it's paid. So you could apply, nearly everyone who wanted to go an internship overseas could apply for this scholarship. And to be honest, not too many people in first year of master wants to do the internship overseas, so you have quite high chance to get the scholarship. And I wanted to improve my English, because I was very bad in English. As you may have As you may know, if you have visited France, French people are very bad in English. So I was like, okay, let's go to America. But I couldn't go to America because you need to pass a specific English test to go. So I was like, no way, I'm not going to get that. So I was like, oh, Australia. They did not require anything, you know. So I said, that's great, let's go to Australia. And I've been to Australia, so you know, I would recommend to check the country and things that there before going, because me, I was like, okay, let's go, and I didn't check anything before going. But yeah, so I would definitely, you need to be prepared if you go overseas and you have never been in a so far country, because it's more than 30 hours of travel from France to go to Australia. So when I arrived in Australia, it was like amazing. It was an amazing internship. So what people say about Australia is when you arrive, usually you cry because it's very hard to get there. You know, it's a journey, and especially when you are 23 years old. And then when you leave, you also cry because it was such amazing and you do not want to leave Australia. So I really like my master 2 months internship. And when I was there, my PI told me, oh, would you like to come back to do your PhD? And I'm like, sure, but I don't have the money to pay these enormous tuition fees, because in France, it's free. You basically pay 387 euros per year for your health insurance. That's it. So in Australia, it's 20K, you know, per year. So I was like, I don't have 20K to pay for my tuition fees. So my PI say, wait, you have done some research work during your master internship. So let's apply for a scholarship. And I applied for a scholarship, and I waited 1 year because I was back in France to do my second year of master. So I did my second year of master at the University of Poitiers. So this is another university, because my PI told me you need to specialize in genomics to come back for your PhD. And I was like, okay, sure. So I did a master molecular biology and genetics at the University of Poitiers. Then I had a 6 months internship, research internship at the University of Limoges in a hospital, in a research lab at the hospital. And then while I was doing my internship, I received the great news that I got 2 scholarships to do my PhD in Australia. So I got a scholarship to pay for my PhD, so every month you get a salary, and then I got a scholarship to pay for my tuition fees. So I would say, like, make sure, guys, when you apply for a scholarship, you get both, you know, because unless you have money to pay for your tuition fees, but it's important. I've met some people in Australia that did not realize that needed all this money to do a PhD, and they basically gave up, and they were back in their home country. So I did my PhD on the human Y chromosome, the long non-coding RNAs, and coronary artery disease. I will explain you later what it is. So I really liked my PhD in Australia. So this is all the things I basically did in Australia. They are in Australia, so they are like amazing place to visit. I really encourage you to visit Australia. The nature, the wildlife is amazing. This is Aboriginal arts. Here it's in the north of Australia. There is nothing to do, so kids are actually, this is termite mound, and people put clothes on this. This is the Great Barrier Reef. I was able to see it. That is unfortunately like dying. Those were the birds in my garden. So all these pictures are mine. This is like also in the north of Australia when I saw crocodile. There are also like very, very huge bat. I don't know if you are aware about that. So, you know, in Australia we talk about like spiders, snake, but there are also like very, very huge bat. And there is a place unique in the world at the top north of Australia where crocodiles eat bats. This is a unique place there. Unfortunately, when I was there, I was not able to see that with my eyes because there was a TV that was like filming that and we couldn't go, but yeah. And this is the Daintree Forest. This is the oldest humid forest in the world. So this is in top east of Australia. I also fell in love with koalas. I didn't know I was liking koalas, and when I saw one, I was like, oh my God, this is amazing. Here, it's a famous place also in the north of Australia. This is the age of the territory, and here, this is they call that the Arnhem Land. This is the land for Aboriginal people, so no one can go through you're not allowed to go on the territory, Aboriginal territory, and it's an amazing view when you are at the top of the hill. And here it's like the end of my PhD. It was like when I was writing my thesis, I was thinking, oh, maybe it's worth a picture. It's my desk. It's a big mess. And that is when I received like the book. They print the book of your PhD. And that was like in my lab. So I really, really enjoyed my PhD there. And after Australia, I had the opportunity to apply for a postdoc here in San Diego. And I was thinking, no, I will never get the position. And yes, I got the position at La Roya Institute for Immunology, where I studied monocytes and long non-coding RNA. So I would never thought I would ended up in America, and here I am. So this is all the opportunities I was not afraid, to take during my background, and I'm very happy to be here. Also, like this small picture here, you might have recognized this is the Y chromosome and the X chromosome. So just for the story, the Y chromosome is decreasing time to time, so this is why it's very small, and at one point, but don't worry, like in a very long time, it will disappear. And so after my short postdoc here in La Roya Institute, I ended up at the stem cell genomics core at UCSD. Again, I will never thought I will be at this position, and I really, really enjoy the position. So being in the core is actually you are responsible of a lot of very expensive instruments and technologies, and you are helping other scientists and researchers to use those technologies. So thanks to this position, I was able to be trained with the brain, like cutting-edge technologies that we have, and we are also very lucky, like here in San Diego, like all the new technologies are, are born, most of them are born here, like in California. So I've learned a lot about genomics, and it's really like, I figured out that I really like genomics and molecular biology. So this is like quite a project we had. Here is actually a scientist and a doctor, MD, PhD, that was here from my university in Limoges, and we were studying a human artery. So he came here to do like an experiment at UCSD, and here, like both cells, this picture is for me like, remind me like very important things, because at that time when I took the picture, we were checking if cells that had been to the International Space Station were still alive when they came back on Earth. And yes. So now let's come back to here at Salk Institute. So this is our building where the NGS core is. So actually, it's in this building. So you might have not recognized this building because today it's raining, so this is not the same picture. And to understand what I'm doing here at the next generation sequencing core, I need to explain you what is DNA. So you might know already, so it's desoxyribonucleic acid. He has a double-stranded helix. DNA is made of complementary DNA bases, A, T, G, C. The order of those bases, the sequence, are very important because it's what determines your genes. So everyone has the same, approximately the same number of genes, but not everyone has sometimes the same sequence. So we will see that there are some mutations. And the human genome is actually made of 3.2 billion bases of DNA. So if you want to know approximately what it is the total length of all your DNA strands in the human body is 6,000 times the distance of Moon from the Earth. Can you imagine? In all your body. So this is more in details, okay? Let's go in details. So you have the double helix, and the double helix is wrapped around complexes of histone protein here. Is that giving the chromosome a more compact shape to be able to fit into the nucleus of your cells? And basically, the human body is made of tissue, tissues are made of cells, okay? So all your DNA and chromosome are in the nucleus. And so we are going to watch a video about it.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is to fit into the nucleus of every cell.
Don't have sound here.
The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule.
So as you can see how it's shaped.
The combined tight loop of DNA and protein is the nucleosome.
And it creates a nucleosome.
Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes.
In the cells, so they are not always visible. The chromosomes are visible only.
It is important to realize that chromosomes are not always present. They form only when cells are dividing.
Chromosome here and the cells is dividing into. You see, so all chromosome goes
At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
It was 1 and then now it's 2 cells. So this is like a real, it's in real, okay? It's not an animation. Okay, so let's now. Shoshana, I think, yeah, we need to put back the. I think it's not in presentation mode anymore, you know what I mean? Okay. Yes. So now what is sequencing? So DNA sequencing is a process of determining the order of nucleotides, so A, C, G, T, as we saw earlier, that makes up a DNA molecule. So DNA sequencing allows scientists to read and analyze the genetic information contained within an individual's DNA. By understanding the sequence of an individual's DNA, scientists can gain insight into their health, ancestry, and also potential risk of certain diseases. So again, sorry for that, we have
Hello and welcome to today's talk about next-generation sequencing, which is also known as massively parallel or just deep sequencing. Next-generation sequencing has completely revolutionized genome analysis. With next-gen sequencing, it is possible to sequence an entire human genome in just one day. We start with the sample preparation. Once we have isolated our DNA, we need to generate smaller fragments. Now all of these DNA fragments will get ligated to certain oligonucleotides. There are two types here. These include sequencing binding sites and also an additional complementary sequence which can then hybridize to the flow cell. First, our DNA double-stranded fragment is denaturated. So in theory, the whole genome should be in fragments and all of the fragments have attached two different types of binding sites on each end. All the DNA fragments are now applied on a chip, the so-called flow cell, and as you can see here, there are two types of complementary sequences, the blue ones and the red ones in our example. The DNA fragments will at this ends hybridize with the flow cell as applied to the plate. To make this easier, we just look at one fragment. The first step here on the flow cell is the DNA amplification. By polymerase chain reaction, the complementary sequence to our single-stranded DNA fragment is synthesized. This process, which we have seen here, is done simultaneously for all DNA fragments on the whole flow cell. Now the DNA is denatured and the single strand, which is not attached to the flow cell's oligonucleotides, gets washed away. The next step is called bridge building. You may have noticed that we have two different types of oligonucleotides here, and the second type of oligo will now also hybridize to one of the sticky oligonucleotides on the plate. This process is called bridge building. The DNA is now amplified again, so a polymerase will synthesize the complementary sequence again. This whole process is called bridge amplification. The DNA is denatured, but now you can see that both fragments stick to the plate. The whole procedure will just repeat now. The DNA fragment will build a bridge again, and again we also have the bridge amplification. That whole process will be repeated for several rounds, and this here is the result. So out of one DNA fragment, we generated multiple copies by PCR. We have reverse and we have forward strands from our copy here. One of these strands here is cleaved now, and sequencing can begin. For sequencing, we just look at one copy now. As mentioned in the beginning, a primer can bind to the oligonucleotides, so this is a sequencing binding site here. Sequencing is now done with specific nucleotides. These nucleotides in this case are fluorescently labeled. Only one of them will fit. In this case, the T. The fluorescently labeled T will then be excited by a laser, and the fluorescent signal is obtained. This is done with a color code, so we know that in this case blue is T. In the next case, the A will join. Again, the fluorescently labeled A is excited, and we will receive a signal again, this time an A. This whole procedure is done for all of the nucleotides, one by one, and this is how we get the sequence.
Okay, so now I'm going to show you all the big machines we have here at the core. So we are very lucky we have 4 sequencers instruments. We have the Nova 6000, NextSeq, MiniSeq, so those are instruments from Illumina, and we have also like a Sequel 2 PacBio instrument, which is not Illumina, it's PacBio. So those 3 from Illumina allows to read and sequence would say like normal length of DNA and RNA, and the last one able to read like super long, we call that read. So if you have like a specific gene that is very, very long, or a specific RNA that is very, very long. And then we have instrument to study within the cell, so at a single cell level. So the instrument on the left allows you, so you take your cells and it separates all your cells into one single cell in a lipid droplet. And then you can sequence each lipid droplets and the DNA or RNA that is inside, and to read what is in your single cell. So for example, it's quite used for cancer research, because as you may know, sometimes in cancer cells, you have specific cells that have a different mutation. And sometimes, for example, when you have a treatment for cancer, you kill most of the cells, but some cells are still alive and they mute and they create another cancer after that. So we usually use the single cell technology, mostly in cancer. And then we have another instrument, the isoplexis, that is also for single cell. So you put your cells in this mini incubator and it's going to record in live how many proteins and what type of proteins your cells are producing. And then we have spatial transcriptomics instrument. So spatial transcriptomics is a new field of research, and it allows you on a tissue to see directly where your genes are, okay? So instead of like mixing all your DNA or RNA together and then sequence your DNA or RNA, we are going to put like, for example, a piece of tumor here and study exactly what is the transcriptome or the proteome of this specific type of tissue. So maybe like this part is completely like, you have like a lot of gene dysregulated or mutated, and here it's quite normal. So this is very interesting also in cancer or specific type of diseases. And this is another technology, so from a different company, but it does the exact same thing. So here you have like a brain, a tissue brain, and you can see under each dot the different expression of genes, especially in the brain, in a tissue brain. Okay, so now you're going to answer the question, what is the purpose of studying genomics after everything that I told you? So just to remind you, genomics is an interdisciplinary field of biology focusing on the structure, function, evolution, mapping, and editing of genomes. So basically everything that is related to the genome, how it works. So basically we are studying DNA, but also RNA, protein, everything that is in the cells and in the tissue. So you can, are you answering right now? Yeah, okay. Yeah, I think it's pretty good, guys. You got it. You passed the test. Okay, and to finish, I just would like to mention Francis Crick, who was a PI here, because this month is the 70th anniversary of like the discovery of the DNA molecules. So indeed, on February 28th in 1953, Francis Crick said, I have discovered the secret of life, which was the DNA molecule. And he proposed the central dogma, so here, that I wrote here, so this is DNA are converted into a functional product that is RNA and then protein, and that was proposed in 1958 by Francis Crick. Today, we know that actually this dogma happens for only 2% of our genomes. So 98% of our genome is not this. So 98% of our genome doesn't make protein. And so at that time, people were calling the 98% junk DNA. And later on, people are starting to figure it out. Oh, okay, it's impossible that 98% of our genome is junk. So they studied this and they find out like a lot of RNAs that were not coding for proteins, so we call them the non-coding RNAs, had actually a function. So for example, you may know that women have 2 X chromosomes and men have 1 X and 1 Y. So actually one of the X chromosome in female is silenced. So no genes are expressed. Because it's normal. If your 2 chromosomes were expressing all the genes, you would have an imbalance compared to men. So the process of 1 X chromosome that is silenced is actually made by a long non-coding RNA, so a non-coding RNA that's called cyst. And so that was one of the first one studied, non-coding RNA. And so I did my PhD on long non-coding RNAs, which are like specific part of the non-coding RNA family. So this is, you know, like this is only 2%, and the 98% is quite a large field of research that I would encourage anyone to study. And we will finish by all these scientists here at Salk that are working. So as you can see, there are different fields of research, what they do every day. So you have like plants, you are also like working with RNA, DNA, microscopes. You're also working with live cells. So here it's a cell culture room where people culture cells here. And we have also a lot of robotics. And here, bacteria. So thank you very much for your time today, and I hope you learn a lot through this presentation.
Turn it on. Everyone have attention. So now we have time for some questions, and there's a couple of different options that you can answer. If you don't want to raise your hand, which you're welcome to do, you can raise your hand and ask your question live, or you can go to Mentimeter and put your question in. And you can ask Dr. Molina anything about her journey, advice, or anything about her science. So please feel free to ask her anything about her presentation, any clarity. I know my question was, I didn't get to see the Mentimeter. What was the answer for what is the purpose? What do we do with sequencing? Oh. That was my first question.
Yeah, there was several choices. I don't remember if you put several choices or if they had to write. Okay, so the purpose of sequencing is actually reading your DNA or your RNA and find out if there are some mutation or abnormalities in your genome. So of course to see disease, problems, epigenetics, all these things. So you understand why now sequencing is important? Yeah. So I think we have a question about yeah, because it's here. Yeah. So someone asked if the X chromosome is silenced. No. So in men. So in men, the X chromosome is not silenced, so you're expressing all of your genes on the X chromosome as well on the Y chromosome. It's just on female, you have only one chromosome, one X chromosome that is expressing of the genes. And then you have why will the Y chromosome disappear? So that we actually don't know. It just is like is smaller than the others, so is losing some genes. And so scientists have calculated that statistically is going to disappear at one point. And we have, what did you do to earn more motivation in your journey? I'm not sure. I was just motivated by everything that I was like studying, seeing, like living. So I didn't do a specific change. I was just like taking opportunities when there were opportunities opening in front of me.
Okay, and while she's looking, is there anyone that has a question that they want to ask live? Great, we have a mic right here.
Okay, guys, you have a lot of questions.
Sorry, I'm just a little bit confused on the Y chromosome disappearing. Do you mean in the individual or as a whole?
As a whole.
And what's the estimated time for that happening?
Oh, like, don't worry.
Oh, okay.
Yeah. We are talking about like millions years. Yeah. But yeah, it's true. It's true. It's true. It's happening faster than other chromosomes. You know what I mean? Yeah. It's quite specific to the Y chromosome. So scientists are believing that at that time, the female will have to find a way to reproduce by themselves without the Y.
We have one more question from the audience.
So no Y, no males?
Yes. Exactly.
Is that possible? I mean, I don't know.
Yeah, some people are looking for that day.
We hit on a hot topic here. Do you have more online?
So someone is asking me what I have having fun studying here at Salk. So at the core, you are studying everything. There are plant, cancer, like every species. Recently, for the first time, we worked with squid. Yeah, that's my first time. So that was.
We have one more question from the audience.
If one of the X's is silent, then why do sex-linked traits occur 10 times more often in males than in females?
Okay, can you repeat what? Sorry.
Sex-linked traits like colorblindness or muscular dystrophy or hemophilia that occur 10 times more frequently in males because they only have one X. If one of the X's in females is completely silent, then they really only have one functioning gene.
Yes, it's because some of the diseases are linked to sexual chromosomes, and some other diseases are not linked to sexual chromosome. So, for example, coronary artery diseases happens more often in male than female, but people are still figuring out if it's linked to some genes on the Y chromosome or not, okay? So imagine if a disease is linked to a Y chromosome, why some female also have this disease, you know? So we still don't know, you know, we are searching. But definitely there are some diseases where there are more male or female that get this disease and it's not linked to sexual chromosome. So some people might think that, okay? Because there are more male with this disease or more female, so it might be linked to sexual chromosome. It's not true. That is not true. It was your question?
No.
No?
So the sex-linked traits,
Yes.
like colorblindness,
Yeah, okay.
that's on the X,
Okay.
and females have it 1/10 as often because they can be carriers.
Yeah.
Because one can have the defective gene, one can have the normal gene, and so that female is a carrier,
Yeah.
but she can give it to her offspring.
Yes.
And if a male gets the X from his mother, then he has color blindness.
Yes.
But her daughters usually will be carriers because the X from their father will not have the color blind. So I'm not understanding the silenced part.
So that I would need to sit with you and to draw, because like every gene has two alleles. Okay, so it's linked with that. Also, when, for example, if a couple have like a girl, okay, this girl is received the X chromosome of the father and the X chromosome of the mother. Okay, it's not in all your cells. It's not the same X that is silenced.
Okay.
It's by chance. Okay? So for example, if you think your X is A, you have A and B, in some cells, A is going to be silenced. In other cells, it's B.
Okay.
Okay? So this is why we also call that like the penetration of a disease. Some female might have the same disease, but some female have the disease is more important than another female. It's just because of that. The mosaicism, we call that the mosaicism of the X, because maybe like the mutation is on the chromosome A, and most of the cells in her body, the A is silenced. So she doesn't have, much of the disease. It's not very important. But for example, like another female, the mutation is on A, but it's most of the B in her body, the chromosome B is silenced. You know what I mean? It's very complex. So I should draw you things, it's hard to explain like that.
Okay, we might want to check. I'm moving.
Yes.
I think we have a few more coming in online. Does anyone else have a question live? Okay, we have one more from the audience.
The favorite part of Australia definitely was the North Territory.
We have one more from the audience.
So I think there's a kind of generality about having to go to a really prestigious college to get a really impressive job like this. So would you consider the colleges that you went to prestigious because they're out of the country and I don't really know other prestigious.
Okay, sorry, you have to talk more. Yeah. No, so I thought like you, but it's actually no. So I really think it's the work that you do, you need to be passionate, you need to work hard. But I'm lucky it didn't play really like a role because the university I've been in France, they are not on the top of the list of the best universities in France, not even, so don't think even in the world. The university I've been in Australia was quite new, so it's not listed yet, on the top university because it was quite new when I started. And so, I don't think, of course, I think sometimes it plays a role to be in coming from a prestigious university, but I really think here in America, they see what, where you are capable of and what you have accomplished so far, as work, not the schools. No, I never. Yeah. Has there been a time in your academic journey when you question being in a STEM field? So no, never. No, I never thought, like I'm a female, I should not work in science. No, I never thought about that. I think I was lucky all over my career. I never been in a situation where someone was judging me because I was a woman or because I was coming from overseas. No.
Okay, any other last minute questions from the audience? Okay, we've got time for one more.
Yeah, I've seen someone is asking about hermaphrodism. That is quite complex regarding genetics. So it's different. So you have sexual chromosome abnormalities that is different from what you are feeling. You know, if you feel that you are a woman or a man, that is different that the genetics. So I'm happy to discuss that later, further with the person who asked this question. But there is some sexual chromosome abnormalities. So some people can be born with, instead of having one X and one Y, for example, they have X, they have two X's and one Y. Or some people also can have two Y's and one X. So yeah, I can discuss that further with the person. Yeah, so I cannot also recommend some state colleges here in America because I've been here in America for 6 years, and so I don't have the knowledge, so I cannot tell you about here in America. Yeah, so I think we are done, guys, so I'm happy to answer any more questions later on during lunchtime, for example, or during breaks. So what I would like you to remember is always do what you like to do, in the future. Don't stress about too much about the future, and you have to be happy in what you are doing. It's very important, otherwise you might give up, drop your class, etc. So it's very important you are happy in what you are doing. Thank you.
Thank you, students, and thank you, Elsa. That was amazing. So we did get a couple more questions on there that weren't able to be answered either by time or weren't really in Elsa's expertise. So if you do have questions, let's say about how Salk is funded or the way that it's run, I'd be happy to talk with you about that more during our lunch hour. So during lunch today, we will have plenty of scientists and Salk professionals for you to talk to. Anyone that is wearing one of these shirts, they are a Salk person that you can go and talk to. So I highly recommend that you go and ask them questions. We're going to be going over a little bit right now of what we are actually going to be doing today. So I'm going to open up that slide again. You're welcome to sit down if you want to. Yeah. Okay. Okay. If you can hear me, clap once. If you can hear me, clap 2 times. See, this is why I love having students here, because they still know how to do that. Scientists, they lose it somehow along the way. They forget how to do that. So today we are going to do a lot of fun things. So you guys are actually going to be going into actual labs here on campus. You're going to be participating in some demos, some lab demos. You're going to be seeing what it's really like in an active research lab here at Salk Institute. And you are also going to be participating in a micropipetting workshop. So micropipetting is a really important skill here in all areas of research. So we're actually going to be doing some training times with all of you. Now, if you can take a second, look down at your name tag. So you might have put it on your hat, you might have put it on your shirt, on your leg. All of you should have a number on your name tag. Teachers as well. So teachers, if you don't have a number on your name tag, please check. So, gonna wait again. We are gonna be going into labs, but not everyone is going into the labs at the same time. Your number is going to dictate where you are going. In just a moment, we're going to go out into our foyer again, so that big open room with the tables, and you are going to find the person. They're going to be holding a stick with your number on it. You are going to go to that person, and they are either going to take you to a lab or they're going to take you to a micropipetting workshop. The odds, so if you have an odd number, you're going to be going to a lab. If you have an even number, you're going to be staying for a micropipetting workshop. And then around 10:50, we're all going to switch. So everyone's going to get to do both activities. As you can see from our schedule, we have lunch at noon. Students, I know we're sitting down for a long time. It's a Saturday. Give me a few more minutes and then you'll get to run free. So at noon, we're gonna have lunch. We're gonna have a testimonial booth upstairs. So we wanna hear from all of you what your experience is like in the lab. So we wanna hear from you of what your experiences were today. That's gonna be upstairs. We're also going to have a career panel at 12:30. So at 12:30, we're going to have a career panel. We're going to have some Salk professionals up here where you actually get to ask them really thoughtful questions about what it's like to be here at Salk. And they're not just scientists. We also have someone from OTD, so they work on patents. And we also have one of our scientific illustrators. So we have an amazing career panel later today. When you go into the labs, so in just a moment, when I excuse you to go back into the foyer and find the people with the stick with your number on it, when you go into labs, please do not leave any food or drinks, or don't take anything into the lab. It's just your body, okay? So don't bring anything in there, don't leave anything in there. So if you have your cell phone on you, make sure it stays with you. If you are touching anything in the lab, please put on gloves and then wash your hands afterwards. These are active research labs. So there's going to be stuff on some of the counters that are not good for getting into your eyes or your mouth. So please make sure to put on gloves and wash your hands afterwards. The lab will provide all of that. And then at the end of the day, so after all of this, we're also going to have some surveys for you. And if you complete the survey, you get some free swag. So yes, we love swag. Okay. Now, what we're going to do right now, we're going to give one final round of applause for Elsa, and then we're going to go into the foyer.